Okay, hi everyone, I'm Lincoln. I'm a grad student in Bill Noble's lab in the Genome Sciences Department at UW. And today I'll tell you about imputation of cancer proteomics data with a deep model that learns from many data sets. So hopefully that works, yep, okay. So to motivate this, missing values are a challenge for mass spectrometry proteomics. Mass spec instrument technology has improved drastically in the past several decades. We've gotten way better at reliably quantifying certain peptides. That said, missing values are still a problem, especially for certain acquisition strategies such as TMT or tandem mass tag, which is what I'll focus on today. Because of the sort of stochastic sampling nature of TMT acquisition, you're always, well, maybe not always, but like for now at least, missing values are certainly a problem. So to define these real quick, I'll say that peptides, so missing values are peptides that are present in the sample, but don't necessarily have an associated quantitative value. So that's what we mean by missing. These are a problem because they can reduce statistical power, they hinder reproducibility, they make it difficult to analyze low abundance peptides and proteins, which are often super important in the disease context, for example, cancer and neurodegenerative disease. And it's also worth noting that they're often missing not at random. That's to say that low intensity peptides are much more likely to be missing. And to briefly illustrate this point, I have data from this 2019 MCP paper in which they're looking at peptide missingness fraction as a function of the number of TMT plexes that you have in your experiment. And so the takeaway here is that, I wish I had a laser pointer, but is that, if you only have like 1 or 2 plexes, missingness is maybe like less than 20%. That's probably manageable. But if you have an experiment with like 20 plus TMT plexes, missingness is now like 50%. And that's, we should do something about that. So like I said before, this is especially a challenge for these like large scale multi-batch TMT experiments. One example of which is CPATAC. So this is the Clinical Proteomics Tumor Atlas Consortium project. It's this large scale multi-site study of 10 different cancer types, which are shown on the left and are abbreviated. So they looked at over 1,000 different patient samples from 10 different types of cancer. This was a multi-study center and multi-center study in the sense that there were 3 different data collection and processing centers in the US. Samples were run by different experimenters, of course, on different days, even on different instruments and different instrument platforms. So I think it's okay to think of these as separate data sets. And what I'm showing on the right is the percentage missingness for each of these 10 CPATAC cohorts. And so the missingness percentages here are quite a bit lower than on the previous slide. And the reason for that is because here we've rolled up to the protein level. And so when you do protein level roll up, you are masking effectively a lot of your missingness. And so you should expect it to be a bit lower. Okay, so this brings me to imputation, which we can define as using statistical or machine learning procedures to estimate missing values. This sort of thing has been done for a long time in genomics. It's sometimes referred to as data denoising. They're not quite the same thing, but they're very similar for folks with genomics backgrounds. There actually are quite a few methods for doing this for proteomics. However, they are largely borrowed from microarray analysis. That's to say there aren't a ton of them that are like specifically designed for the specific, data needs of mass spec proteomics. There's only one that I know of that uses deep learning, and none that I know of are able to learn from like multiple data sets at the same time, which has like sort of become a paradigm of modern deep learning and machine learning. And then on the left side is shown a figure of citation counts for the most commonly used imputation methods in proteomics. So what you'll see is that Gaussian random sampling is by far the most commonly used one, and that's just because this is what Perseus does, in spite of the fact that this has been shown to not work super well. Okay, so what we did is we developed this method that we're calling Lupin, which uses deep matrix factorization to impute missing values in TMT proteomics. So right now we're focusing just on TMT, but in the future, hopefully we can scale this up to other acquisition strategies. So the first thing that we did is we created this, what we're calling a joint quantifications matrix. So here we combined runs from all 10 CPATAC cohorts. And so the different colors here correspond to different types of cancer, different CPATAC cohorts. Every column is a sample, so a patient sample, and then every row is a protein. And we just join these together on shared proteins. And if a given patient sample doesn't have a protein, it just gets a value of nan, so not a number. And then here I'm indicating that we also withheld, say, 20% of our data in this matrix as a test set. So that's data that the model does not have access to during training. The next thing we did is we factorized this matrix using non-negative matrix factorization, which gives us these two product matrices that we're calling protein and run factors. And then for a given missing value, for example, the one that's shown in light blue, maybe it's hard to see without a laser pointer, but for a given missing value, what you do is you take the corresponding protein and run factors, you concatenate those into one long vector or perhaps matrix, and you feed those into a deep neural network. And that gives you a single prediction value for that missing value. And you can iterate on this until you have protein factors, run factors, and then deep neural network parameters that are giving you really robust and accurate predictions for your training set. And then you can say, okay, for my test set, how well do I do, or how well do I do at reproducing the values in that test set? And this is to simulate the setup that a user might have in which you have missing values for which you don't have ground truth, and you want to know, like, okay, what's a reasonable estimate of how accurate I'm doing at imputing those? And so the result shown here is a comparison between Lupin, our model, and Dream AI, which I think is the current state of the art for TMT imputation. This is actually a project that came out of the CPTAC consortium itself. It's an ensemble imputation method. I think it works very, very well, generally speaking. And then Gaussian random sampling on the left, which again is what Perseus is doing. And so every dot here corresponds to a different CPTAC cohort. So there's 10 dots in each of the plots. And what we're showing is that our test set error is lower for each of those 10 cohorts than the competing method. So I think this is a pretty good sign. This is a very quick talk, so I'll skip to my conclusions and just say that missing values, still represent a challenge for mass spectrometry proteomics. We've created this model that we're calling Lupin, which uses deep matrix factorization. It learns from multiple data sets at the same time. We think that we are outperforming the current state of the art. And then one really cool thing that I wanted to touch on briefly is that our model actually learns this pretty interesting representation of proteins and mass spectrometry runs within its latent space, which is shown on the bottom left. So we're able to learn the differences between all 10 cohorts, as well as differences between tumor and tumor-adjacent normal samples, without giving the model access to any of that metadata. So the model is implicitly learning these relationships without actually being given that data. So I think mining this latent space for useful information with regards to cancer biology could be a really interesting follow-up, as well as potentially expanding this to other data modalities. The really cool thing about CPTAC is that you have patient-matched proteomics, phospho, DNA methylation, whole genome sequencing, all kinds of data for the same patients. And so expanding this to include some other modalities might give you a really cool multi-omics model. And that's it. So here's my acknowledgements, and I'll take any questions.
A very nice presentation.
Thanks.
I'm assuming that your model requires a certain number of replicates in order to perform decently well. Have you looked at how many replicates or how replicated the data needs to be so that the model performs properly?
Yeah, so that's a good question. I guess the answer is we haven't looked at it because CPTAC, which is the only thing that we've trained it on so far, just has a bunch of replicates.
Right.
So yeah, I guess we got lucky in the sense that our training data set already has that baked in. It would be interesting to try this on a data set that doesn't have replicates and see how well we do.
Or you could censor, basically remove some of the data.
Right, we could downsample.
See how far you can go until it falls apart.
Yeah, so we haven't tried that, but that's a good idea. Yeah.
Thank you.
Thanks for a nice talk. So the input to the model, is it the raw TMT ratio count, or is it normalized to some degree?
Right. Yeah. So it's been normalized. So CPTAC has all of the TMT plexes have a reference channel, and so everything has been normalized to that reference channel. Yeah.